It has been a passion of mine to really help patients with C. difficile infection and recurrent CDI ever since 2010 when I was in the young lab at University of Michigan. And my team is really committed to finding better therapeutics to combat this disease. So go to the right. So I first wanted to start a little bit about my lab, and as Christian introduced it, the Riot Lab or the Theriot Lab, we're really focused on targeted manipulation of the gut microbiota and the metabolome, the small molecules in the gut, to alter GI diseases. And my current team members are shown here on the right, and really these are the people that do all the beautiful work. Without them, it would not be able to be done. And I've highlighted Dr. Abigail Gantz, who's a talented postdoc in my laboratory, where most of the data I've generated today is from her. So in my lab, we use a combination at the top of in vitro, so that would be in the test tube, in vivo approaches, which is in the mouse model, to study C. difficile infection. We also combine this with omic approaches to characterize the gut environment, and we work on a number of translational studies with collaboration at UNC Hospital. And the goal really is to develop novel microbiota-derived therapies to treat CDI. And this approach is also being used to study other GI diseases, including collaborations with IBD at UNC CGIBD, the Center for CGIBD. So we've seen this slide before. This tracks back to Vince Young. We know that antibiotics are important for treating bacterial infections, and I'll kind of go quickly through this, but I really want to focus on the physiology of C. diff and what the actual bug needs and the pathogen needs. But we know that the antibiotics also decrease colonization resistance to where C. diff can colonize the gut. We do know a lot about C. diff life cycle and physiology, which starts at the C. difficile spores at the top right. C. diff is a spore-forming organism, and this dormant state is thought to aid in transmission. The spore form of C. difficile is dormant until it recognizes many germinants, including bile acids, which we've heard today, which initiate outgrowth of vegetative cells. The vegetative cells require growth substrates, so they need food just like us to grow. And there's many amino acids. The ones listed here are the ones that we know C. difficile requires. So it has to have those 6 amino acids in order to grow. It's during this time when you get the high cell density, it grows to high levels, that C. diff produces 2 toxins, A and B, which causes CDI. Available treatments for CDI include more antibiotics like vancomycin. And again, we've heard today of some new antibiotics, hopefully in line 2, but there's a high rate of relapse where patients can go into the cyclical pattern, and that's where the FMT is warranted. So regardless, there's still risks to using FMT, as we really do not know what is in stool. Novel defined therapeutics that do not alter the microbiota, but target C. diff are urgently needed. And one of the major projects in my lab is really to focus on FMTs as we are trying to figure out mechanistically how they're able to work and clear recurrent CDI. Because if we can figure that out, we can actually develop more targeted bacterial therapies and move away from FMTs or refined FMTs approved by the FDA. So in 2017, with the help of NC-TRAC's funding, we received a grant to collect stool samples from patients before and after their FMT from UNC Hospital. Since then, we have enrolled about 30 patients, but today I'm going to tell you about 15 patients stool before and after their FMT. The goal was to understand how the FMT was able to clear recurrent CDI so quickly. What is the mechanism? In order to do this, we defined both the microbiome, the metagenome, and the metabolome of these samples in pre and post FMT samples. First, we wanted to quantify changes in the diversity. And you saw a great talk from Vandanta. So again, and others that have kind of talked to you a little bit about diversity today. So on the left, we have a measure of alpha diversity using Shannon. And before the FMT in orange, we have significantly lower alpha diversity than every other point post-FMT. So we know that increased diversity is associated with clearance of CDI, or recurrent CDI after FMT. We then looked at beta diversity on the right, which measures how different each sample is from each other. And here, points that are closer together are more similar, points that are further away are more dissimilar, and you can see that the pre-FMT group clusters. So we kind of know this. So this was nice to say that our study was very similar to others out there. But we wanted to understand who was present. So which bacteria were present in the pre-FMT and the post-FMT samples. And here on the bottom axis or the x-axis, we have the pre-sample and all the post samples. And what I want you to notice is the relative abundance of that blue bar in the pre, which is those Enterobacteriaceae that we really see present high abundance pre-FMT. And then the post-FMT samples, that goes away, and you're really left with these poo-poo brown colors. And I made them that color for a reason, because they're important. And they're lacnosparaceae, which we'll come back to them later, because they're going to be part of the star of our show today. So what does the metabolome look like? So then we wanted to do metabolomics, and we performed a random forest analysis. And this time we have metabolites along the y-axis, which are colored by the type of metabolite. And so on the left side, we have the list of metabolites, we have which grouping they come from, and if they are a higher MDA accuracy on the right, if the dot is more closer to the right, it means that they're more important in discriminating between a pre and post FMT sample. On the right, there's a heat map where if it's dark, it means it's an increased abundance of the metabolite, and if it's light, it means a lesser abundance. And if you look at the top part, we see in the bottom part, all the dots, we see a lot of yellow, and that yellow indicates lipids. So lipids were one of the main important metabolites that we identified, and specifically acylcarnitines, which are all increased in the pre-FMT, and they pretty much go away in the post-FMT gut. And then bile acids, which is, I've been working on bile acids for a long time. We see a lot of those primary bile acids pre-FMT, and they go away in post-FMT. We see a lot of the secondary bile acids. Today, surprisingly, I'm not going to talk to you about bile acids. I'm going to talk to you about acylcarnitines, which is a new signature and something that we're looking at here. So from this paper, our conclusions were post-FMT, we had a change in the lipid profile. We had high FMT, in pre-FMT samples, we had high acylcarnitines. These went away post-FMT. We had high conjugated primary bile acids pre-FMT. Post-FMT, we had all deconjugated secondaries. And then we had high Enterobacteriaceae pre-FMT, and post-FMT, we had the replacement of lacnose. Something else that came about this study was that in these Enterobacteriaceae, we saw that they had genes that were very important for amino acid biosynthesis, the same amino acids that actually C. diff could grow. So we put out this hypothesis that maybe Enterobacteriaceae was helping C. difficile. But before we get into that, we really wanted to go back and look at this strong lipid signal. We wanted to do a deeper dive into which lipids were in these samples, and we asked the following questions. How do these novelly described lipids, because you saw those yellows, change during FMTs? Can any such changes be attributed to taxonomic or functional drivers, other bacteria present? And are these patterns involving lipids that could be inhibitory or actually help C. difficile in the gut? So we went back to our collaborator, mass spectrometrist expert and lipid expert, Dr. Aaron Baker and Dr. Zhang from UNC, and they were able to create this library, lipid library of 877 unique lipids. No one has done this around the world. There's only a few people that can do this. And they went back and looked at our samples a little bit more deeply. And so if we examine the overall lipid compositions of the samples, we find that there are significant differences, again, associated with sampling time with the pre and the post-FMTs, with pre-FMT sampling becoming more and more different from the post-FMT samplings when we're just looking at these lipids. The next question is which lipids were actually changing. And to answer this, we use a differential abundance analysis. And in this volcano plot, I've only highlighted the ones I want you to see. And on the x-axis, it represents the effect size. And on the y-axis, it's just the significance. Each point represents a lipid species, and the ones on the left are associated with pre-FMT, while the ones on the right are associated with more importance in the post-FMT. The two types of lipids that emerged as the biggest drivers of pre and post-FMT are acylcarnitines, which I talked a little bit about before, in yellow on the left, and ceramides in light pink. And I'm only going to talk to you about the acylcarnitines today because we are strapped with time. So what are acylcarnitines? They're fatty acid derivatives formed from carnitine and acyl groups. They're key intermediates in fatty acid transport and into mitochondria beta oxidation, very important for host health. They're categorized by their chain length. They can be anywhere from short to medium or long chain. They also are really important because they're indicators of altered metabolism in many diseases that are listed here. And they're both host and microbiota derived. And that's interesting to me because I'm a bacteriologist, microbiologist, and I want to understand what these bacteria, what enzymes are actually altering them, et cetera. So what we noticed when we looked at these acylcarnitines is that the ones on the left side, not in the black box, are medium-sized acylcarnitines, and they're all increased in the red boxes in pre-FMT, and they all decrease post-FMT. And the only inverse relationship is a long-chain acylcarnitine, which is AC26, which is in that black box, which has the inverse, and it's low in pre and high in post. So the next thing we wanted to do was we wanted to see which bacteria these were actually associated with. So we took we saw that the Enterobacteriaceae were positively correlated with these medium chain acylcarnitines. And in the red box, all the red color means that there's a positive association with that bacterium to that acylcarnitine. And you can see that this beautiful, nice positive correlation with Enterobacteriaceae in these medium chains, and the opposite with the long chain. And the long chain acylcarnitine is positively associated with Lachnospiraceae, which I really didn't talk about. So in conclusion for this, basically FMT significantly alter the lipidome of post-antibiotic recurrent CDI. Medium chain acylcarnitines are the strongest drivers of lipidomic changes. And again, we see that Enterobacteriaceae are strongly correlated with medium chain acylcarnitines, and in particular the Klebsiella, whereas Lachnospiraceae are associated with those longer chains. What I didn't show you is that the Klebsiella and the coli from those Enterobacteriaceae, they dominate the pre-FMT, but they also have those genes that are important for making amino acids, the same amino acids that C. difficile requires for growth. And really, this is the new hypothesis that we're chasing right now in the lab and we're testing. And I think it's quite novel, actually. I don't know if it's been put forth before, so this might be new. So we think that the host-derived carnitine and acylcarnitines are actually released during toxin-induced inflammation by C. diff in the pre-FMT gut. And we think that those carnitines Enterobacteriaceae can actually utilize those for growing. And we also think that Enterobacteriaceae can also help C. diff persist and colonize in that pre-FMT gut. At the same time we were doing this, a beautiful study by Gary Wu's group at Penn looked at IBD patients in peds especially, and looked at this acylcarnitine and carnitine increases also in associating with Enterobacteriaceae. And then he found that Enterobacteriaceae could actually utilize these carnitines for growth. So this sets up a really nice hypothesis that we can then go and test. So what we're doing now is we are going from 15 patients to 33 patients. So we're increasing our sample size for pre and post FMT. We also have healthy controls that have nothing to do with this study at all, that are the same age matched, no comorbidities. So we're seeing how healthy looks. We have expanded metagenomics. We have targeted amino acids, lipidomics, and bile acids. And we're looking at C. diff spore enrichment in these samples because we want to see if C. diff is still sticking around. And it is. So to an extent, we have a small study that we've done so far. We still see some C. difficile spores sticking around in post-FMT samples. We want to understand what that is about. We're doing in vitro and in vivo experiments with Enterobacteriaceae to see if they're actually helping C. diff. And then we're also expanding this to IBD patients and Crohn's disease patients with UNC and Penn, because we think that this might actually be very similar and opening up IBD patients to be susceptible to C. diff. So we think it could be relevant in those patients as well. And I'll leave you on my final slide as we continue to interrogate the interactions between the host, the gut microbiota, lipids now, and the pathogen. And on the right side of this slide, I have a huge arm of my lab literally just doing bile acids and trying to understand what bacteria are making bile acids and how that affects C. diff. And then today on the left, my lab is pretty much expanding into these new lipids, acylcarnitines, because we really think that the acylcarnitines liberated by C. diff are helping Enterobacteriaceae, making the amino acids, and then that's helping C. diff. So again, we really want to understand targeted therapeutics so that we can get rid of Enterobacteriaceae, and then that will help potentially get rid of C. diff, or so we think. And with that, I want to thank my lab. I want to thank my collaborators inside and outside the triangle, my funding, and I really want to thank all of you today for your attention and advocacy. And thank you very much.
Well, thank you. Excuse me. You would think after talking all day that my voice would have cleared up, but not so far. Well, thank you so much. That was so interesting. Who knew that fats could do us so much good? We do have a question for you
Sure.
from Jacob, who says, the antibiotics we use for CDI increase lacno and oscilloparaceae, important for restoration, but may hinder engraftment in LBP or FMT and not deplete gram-negatives. Is it crazy to ask if we should try an oral gram-negative antibiotic to deplete Enterobacter cocci based on what you showed?
Yeah, so I think that's an interesting idea. I don't think I have the answer yet because we haven't really proven our hypothesis yet. But I think it's important to understand when you're putting something back in of an antibiotic depleted gut, what's there, because I think it's going to become important if it's going to allow whatever you're putting in, if it's hospitable or not inhospitable for what you're putting in. So I think if we understand the Enterobacteriaceae are, aiding in C. diff or possibly altering one of your LBPs or something, if we can get rid of that and get better engraftment or better responses, then that's going to be important. So I think you have to understand what's in the gut before you put something back in as well.
I mean, that seems fair to me.
That was not very articulate, but yes, I think you need to understand the environment because again, if you put these LBPs back in and the environment is not going to allow them to grow, then your product might not work.
Right, and then that's, the product itself is sort of correct. It's just that it's being put into an inhospitable environment.
Yeah.
Which, we have seen a lot of that with people who, their microbiome just doesn't seem to grow back correctly, and they, struggle for years after C. diff or antibiotics. Or, I don't know if you remember, this was years ago, but there was Marty Blazer had a thing in Nature about how, for like millennials, by the time they got to be 18, they'd all had like 20 courses of broad spectrum antibiotics. And I mean, totally wild, just even compared to our generation,
and they don't recover, right? So eventually they're not all going to recover over that time. So you're going to continually lose that diversity. Very slowly. Yeah. And what does that mean though? So again, we still kind of don't know.
Yeah. Well, so we had one incredibly complex question for you, but that's all we had.